Hello, my name is Trinity Lavenhouse, and I am presenting my research on assessing soil organic matter in a legacy wetland mesocosm after the colonization of invasive species. And my mentor was Dr. Changwoo An. So to start off, I'm going to explain the importance of measuring soil organic matter. Soil organic matter is made up of decomposed plant and animal material. They contain quite a lot of organic compounds and are full of nutrients that help plants grow. So, in general, it is important to measure the amount of soil organic matter that is present because it can indicate how healthy and fertile this soil is for plants to grow. This study was conducted in Dr. On's wetland mesocosm on West Campus in George Mason University. Inside, there are 40 wetland mesocosms that were created in 2012, and the mesocosms were initially planted with 0 to 4 different native wetland species. This picture is from 2014 after 2 growing seasons. Since COVID-19 maintenance halted, unwanted plants weren't removed and the water levels weren't monitored like they used to be. This allowed the colonization of invasive species for the past 3 to 5 years. So the goal of the study was to determine the amount of soil organic matter in each mesocosm after the colonization of invasive species. The hypothesis was that since there were more plants than there were previously, there will be a higher amount of soil organic matter in these mesocosms. To take samples, the mesocosms were split into 5 sections, and one sample was taken from each section within the top 20 centimeters where organic matter is the most present, because soil organic matter decreases the further you go down. This was repeated for all 40 mesocosms. The soil organic matter percentage was processed using the loss on ignition method. To carry this out, each sample was dried at 105 Celsius for 2 days. After they were completely dry, they stayed in a desiccator to cool and were then crushed with a mortar and pestle and were sieved through a 2 millimeter filter to get everything that was too big to go in the crucibles. The fine dirt was then placed in crucibles and fired at 360 degrees Celsius for 2 hours, and they were placed back into the desiccator to cool. Soil organic matter is the percentage of the change in mass. The results were all collected, and all 5 of the samples within each mesocosm were compiled and averaged to represent one mesocosm. It was found that mesocosms that had sparsely spread or no plants had the lowest soil organic matter, while overgrown and dense mesocosms had the highest soil organic matter. The standard error bars are large in mesocosms that had more plants or organic matter in a section than the other sections. The mesocosms were then grouped by the initial number of species planted. There seems to be no significant difference across the board, and the reason for that will be analyzed further. In a previous study conducted by another person in this lab named Mary Means, they measured the total carbon content of all 34 planted mesocosms in 2014. Total carbon content is about 58% of soil organic matter, so that conversion was used to estimate what the soil organic matter content was back in 2014. About 10 years later to now, soil organic matter seemed to increase from a range of 2.36 to 2.59 to now a range of 4.15 to 6.21%. In the future, soil bulk density will be processed to analyze how soil porosity or compaction correlates to the soil organic matter found within the mesocosms. Also, plant identification will be conducted for all 40 mesocosms, as when these samples were initially taken, most of the plants were dead because it was the end of the winter. Thank you for listening.